I'm in charge of number 1. Just read it to everybody. I'm sure everybody's read it, but just for kicks and giggles. Priscilla Lane and Margaret Green studied the relationship between 3 linked genes affected coat color in mice. The 3 recessive genes are mahogany, MG, agouti, A, and ragged, RG. The wild type versions of these are all the dominants. So the scientists carried out crosses between mice that were heterozygous and homozygous recessive. So we got our hetero female here and our homozygous male there. Just doesn't matter who really has what, but anyway. So you all have the numbers in front of you, so I'm not going to go through and read each phenotype of what, how many number of offspring for each one. I'm just going to get right into it. So for A, to determine the allele arrangement, we had to use our parental offspring of each, being 76 and 69. This is the genotype for 76. This is our genotype for 69. This would give us our allele arrangement. To move on to B, we get to figure out our gene order. So we move this down. That's our first gene order. Rotate the M and G and the A. Leave the T. Rotate T and sorry, not T. RG and MG there, or yeah, A and MG, RG and A, and then after that we go to figure out which is our order. So we can find our DCOs, which are 1. This is the genotype for this DCO. This is the other genotype for that DCO. So in order to get these options, we went through each 3 of these and we came out with this one. So this will be our gene order. Now for number C, construct a linkage map showing the distances between the 3 genes. You add up your DCOs, your first SCO, all of the options there, comes out to 13.1 MUs. Again, with our double crossovers and our second set of single crossovers, these are our options. To get those, sorry, I guess I should have explained this. To get our first single crossovers, that's what you're going to do, as well as there, and vice versa for your second set. That's how you get the genotypes for those. After we get this all figured out, second distance for the A to the RG distance is 18.1 MUs. This is our linkage map here. For letter D, we did the math here for our 13.1 over 100 times by our 18.1 over 100 comes out to 0.023711 times by our total number of occurrences, which is 206, comes out to 4.88. This is our our expected number of our DCOs, and then we have to go in to find out the interference. So you do 1 minus the observed DCOs divided by the expected DCOs, all times out to 100, or times by 100 equals 59.02% interference.
Cool. Pretty much you do this for every single problem, but I'll walk you through number 2 just so you understand where all my math is coming from. So, like Tyler said, we set up our trihybrid cross. Since we have heterozygous, we're going to have our dominants on top and then the recessives on the bottom. That's just kind of how it, so if you had it like this, then you would just split those in half, and that's how you get this. And then we gotta find our parental offspring to get the allele arrangement. Your parent offspring is always going to be your highest numbers. So these are our highest numbers on the chart that you guys have there, which gives us the allele arrangement. So for B, we're going to take this allele arrangement and bring it down over here, and we're going to figure out the different gene orders that can go with it. kind of how I set it up. For your first one, you just kind of keep it the same, because if nothing changes. Second one, your first and middle allele will switch. And then your third one, your first, your middle, and your third allele will switch. And then in order to figure out which ones match our DCOs, because that's what we're trying to find, you'll take your DCOs, like Tyler did over here. Use the smallest numbers; those will be your DCOs to get their formations. And you're trying to figure out these and see if these match any of your gene orders that have changed after the crossovers. So it comes out to being this one right here, because you can see it matches here and here. The order doesn't necessarily have to actually match, just as long as the dominants and recessives are in the right spot, then you're fine. So you'll take the original and move it down here to see, rewrite your trihybrid cross, and then use your DCOs and your single crossovers as well to get your MUs. So like Tyler did again, you just take for your first single crossover, you'll just swap your first alleles and everything else will be the same. And then for your third allele crossover is what I think it's called, you just swap the third one and everything else will stay the same. And then you'll look that up in your chart to get your numbers so you can do your MUs. Your MUs is your DCOs plus your single crossovers over the total amount of individuals in your study times 100, and that gives you your MUs. So you do that with the first one and then also with the third allele. So that way you can do your gene mapping up here. Your first allele distance is going to be between your first and the middle one, obviously. And then your second MU is going to be between the middle and the third. And that's how you do that. And then D, you find your frequencies. So your MU over 100, and you multiply those together for both of the MUs. And then you multiply that number by the number of individuals to get the number of DCOs, double crossovers that have occurred. You take that and you plug it into your interference, right? Yeah, for some reason I'm thinking interference equation, which is 1 minus the number of DCOs over the number that you got for the frequency. And you plug that into your calculator and you get 27% for your interference for number 2.
And for number 3, it's basically the same thing again. So we have our alleles up here, we have our cross, our parental offspring. Right here, we have our parental offspring. We do the switching as required to get our different results. We have our DCOs right here. All right, and then for part C, we have our DCOs, yada, yada, yada. Let's see. And then for our single crossover, the first one, we'll just flip the first ones, find our values. And then for our single crossovers, the third one, we'll flip these, find our values. We got zeros for that. It's gonna be important later if we're doing this correctly. And then you get your micro unit values. You get these by adding the DCO values as well as the single crossover values for the first ones. And again, your DCO values and the single crossovers for the second ones. For D, we got our frequency rate. And as we were doing the interference value, or the interference equation, we actually had an error because you're supposed to do 1 minus a smaller number, and that's not what happened. So we have an error. We don't know the actual values. That is all for part C.
Part 3. All right. Unfortunately for you guys, you get to listen to me on number 4. Okay, so the recessive genes, AN for anther ear, BR for bractic, and F for fine are all found on chromosome 1 in corn, when a corn plant that is heterozygous for each of these markers is test crossed with a homozygous recessive for each of the markers, the following results are obtained. You've got your list there in front of you of all of our numbers. So first off, to begin, we're going to start with letter A. We're going to determine our gene arrangement. So we use our parental offspring again, find out what their, based off what they say there, we've got their genotypes. This gives us our gene arrangement. This is where we're going to have all of our genes involved. Then we go to B. B, we need to determine the gene order. So to do so, we write down our gene arrangement and all the different arrangements that it could be. And we're going to go through and we have to figure out using our two lowest numbers, which is our double crossovers. We get their genotypes. Once we have these 6 alleles, we have to find out which one gives us the ability to have all 6 of these in the same area after their crossover. Comes out to this one right here. Gives us our anther, 2 wild types on top, and a wild type and a fine and a bractic on the bottom. Once we've got that, we can move down to letter C to find out and create our linkage map. Using the DCO genotypes again, we find our first set of single crossovers, their genotypes. Using those, we add them all together, divide them by 1,000, then times them by 100. That gives us 19 MUs. Same thing with our second set of single crossovers and our original DCOs. Add them all together, comes out to 5.1 MUs. This is our distance here. We've got from the wild type anther, we've got 19 MUs to wild type fine, and then 5.1 MUs from wild type fine to wild type bractic. For D, again, as you've seen all the way down here on all 3 of the others, we get to find out our frequencies for both, times them together, comes out to 0.00969. Due to our total count number being 1,000, we times that same number by 1,000. Oh my gosh, I can't even write a freaking equation without doing another parenthesis should be right there. Comes out to 9.69. Again, we plug that into our interference equation. Comes out to 48.4% interference.
Alright, so then number 5, don't quote us on this because we don't totally understand it. So for A, you have supposed to sell the genotype up here. Assuming that these are separate homologous chromosomes, what are the genotypes of the gametes that are produced and what is their frequency? So the way we figured that we'd do that problem is you just do a 4x4 Punnett square, and then this is what we came out with, the frequencies for each. Then B, suppose you have the genotypes, and this time they're linked, so that's why we assumed it's kind of like the ones we've done, where they're linked. But we're assuming that the crossing over does not occur. So what are the genotypes and what is their frequencies? So if there's no crossing over, then your alleles is going to stay the same. So that's what we came out to this and having it be 50-50. For C, it's pretty much the same thing. They're going to be linked, except this time there's a crossing over, which gives us different allele arrangements with 50-50 frequency still. And then D, we're given the MU as 7.8, and to find the frequency, we know we have to divide it by 100, and we multiply those and get 0.0061 to get the frequency in percentage. You multiply that by 100 and get 0.61%. So that's what we did.